allies and enemies. Bacteria are the most abundant organisms on Earth. They are everywhere and in vast numbers, up to 40 million in just 1 gram of soil. They are essential for life, but they can also cause grave illnesses. Dr. Hikmate Abriouel at the University of Jaén studies a type of bacteria known as Enterococcus, which are beneficial, but which can also cause problems. Enterococci are frequently found in nature and play an essential role in the production of fermented foods such as cheese and certain processed meats. They are responsible for the aroma and flavor of some of the most highly valued cheeses. They even contribute to their conservation. These round-shaped bacteria also live inside our digestive tracts and only cause problems when our defenses are low, allowing for infections to spring up. The problem is that when this occurs, the infections are quite difficult to treat because they are extremely resistant to antibiotics. They have acquired this capacity from living in contact with these medications inside our intestines. Dr. Hikmate Abriouel and her team have characterized these microorganisms as being resistant to antibiotics and are working to curb their resistance. It is very important to control infections caused by Enterococcus because they can transfer their antibiotic-resistant genes to other bacteria. In order to determine their capacity for resistance, the team isolates the bacteria on slides and observes their susceptibility to antibiotics by adding different concentrations of antibiotics and comparing the results with a control group. In her search for ways to make them more vulnerable to antibiotics, Dr. Hikmate Abriouel evaluated the effects of a product called EDTA, which is used as a food preservative. EDTA is an antioxidant which, when used in the proper amount, doesn't cause problems, and to the contrary, is extraordinarily useful in keeping food from spoiling. In this study, lower concentrations were used than those normally found on the market. But even at these low levels, the effect was drastic. It reduced the minimum inhibitory concentration of the antibiotics. In other words, the minimum quantity of antibiotics it took to kill the bacteria. The cause of the antibiotic's greater effectiveness when used together with EDTA is that EDTA inhibits a mechanism found in the walls of these bacteria through which they are able to excrete the antibiotics. When they are unable to excrete them, a lesser quantity of antibiotics is needed to kill them. Thanks to genetic studies, researchers have shown that this combination of substances inhibits the excretion process, making the use of antibiotics more effective for controlling Enterococcus infections. This discovery has opened up an important line of research for the pharmaceutical industry in the key field of antibiotics and can be applied to other more pathogenic bacteria.